Protein pumps are transmembrane proteins that exist within the membranes of our cells that function to generate electrochemical gradients. So they use some type of energy source to basically move these molecules against the electrochemical gradient across the cell membrane, and that ultimately creates this electrochemical gradient. And then the protein channels, the other type of transmembrane proteins used for transport, can use these electrochemical gradients to basically move the molecules down their gradient without using any type of energy source. So essentially these membrane pumps use energy to establish these gradients, and then the channels don't have to use energy because they use these electrochemical gradients to move the molecules in a natural and spontaneous direction. So there are two categories of membrane pumps. We have these ATPases and we have these secondary transporters. In this lecture, we're going to focus on ATPases. Now, ATPase themselves can also be broken down into two classes. We have the P-type ATPases that we're going to focus on in this lecture, and we also have the ABC transporters. Now, what exactly is a P-type ATPase? Well, the P basically stands for phosphoryl, and in a P-type ATPase, they basically use ATP molecules, they hydrolyze an ATP molecules, they use that phosphoryl group to attach it onto that membrane protein, that pump, and then that creates a conformational change in the pump that allows the pump to actually move the molecule against the electrochemical gradient, as we'll see in just a moment. And one well-studied pump that we're going to focus on, one well-studied P-type ATPase, will be the calcium ATPase that we're going to focus on in this lecture. And there are many other examples of P-type ATPases that we'll study in a future lecture. For instance, we're going to also look at the sodium-potassium ATPase, which is also an example of a P-type ATPase. Now, where exactly do we find, in what membrane do we find these calcium ATPases? Well, remember from basic biology, calcium is used by muscle cells. And so what that implies is the calcium ATPase must be found within muscle cells, and that's exactly right. In the membrane of the sarcoplasmic reticulum, the specialized type of endoplasmic reticulum, found inside our muscle cells, within the membrane, we find these calcium ATPases. In fact, calcium ATPase makes up to 80% of the protein content of the sarcoplasmic reticulum, so membrane. So SR basically stands for sarcoplasmic reticulum. It's the specialized type of ER, which is basically used to store the calcium ions found within that muscle cell. So let's remember how the muscle contraction actually takes place. So our nervous system basically generates this action potential and electrical signal that moves into that muscle cell. And once it moves into the muscle cell, what happens is within the membrane of the sarcoplasmic reticulum, we have these protein channels, not protein pumps, but protein channels. And as a result of the action potential, these protein channels basically open up, and then calcium begins to move down its electrochemical gradient. So inside the lumen of the sarcoplasmic reticulum, so this side, we have a very high concentration compared to the cytoplasm of the cell. And so when the protein channels open up, and these protein channels are not shown in this diagram, when they open up, the calcium begins to move naturally from a high to a low concentration. And what that means is once the calcium moves into that cytoplasm, the calcium can then react with the actin filaments found within our muscle cells that exposes special type of myosin binding sites, and then coupling with ATP, that muscle contraction actually takes place. Now, to relax the muscle, what has to happen is these protein pumps must be able to very quickly and effectively take these calcium and move them back into the inside, into the lumen of the sarcoplasmic reticulum. And when this is done, these calcium ions are moved against the electrochemical gradient. And ultimately, what this also does is it reestablishes that electrochemical gradient that is needed for the protein channels to move the calcium back into this direction so that the muscle contraction, the next muscle contraction, can actually take place. So we see that calcium is needed for muscle contraction, and during contraction, calcium flows into the cytoplasm of the cell from the sarcoplasmic reticulum lumen down into its concentration gradient. And once the contraction is over, to actually relax that muscle, these calcium ions must be sequestered back into the lumen of the sarcoplasmic reticulum. And this is done by these pumps, because this actually requires energy to move these calcium ions against their electrochemical gradient. And this is exactly where the calcium ATPase actually comes into play. Calcium ATPase uses ATP molecules to move these calcium ions against their electrochemical gradient. And calcium ATPase is also sometimes called SERCA, where the S and E stands for sarcoplasmic reticulum / endoplasmic reticulum. The Ca stands for calcium. So the SER stands for sarcoplasmic / endoplasmic. The R stands for reticulum, and the Ca stands for calcium. Or perhaps the C stands for calcium, the A stands for ATPase. I guess that's also a credible way to see it. So we have these calcium ATPases use ATP to pump calcium back into the lumen, thereby reestablishing that gradient. And once this process takes place, we essentially establish a gradient in which, in the cytoplasm, we have a much smaller concentration than in the lumen. And so we have about 0.0001 millimolar in the cytoplasm and about 1.5 millimolar of calcium in the lumen. And clearly, this is a much higher concentration than this. So the pump uses ATP to establish this gradient, and then when an action potential actually comes into that muscle cell, the protein channels not shown in this diagram open up, and because of the existence of this electrochemical gradient, the calcium ions can quickly and effectively move into the cytoplasm where they are needed to basically contract that muscle. So let's take a look at the following diagram. This is basically the generalization of how the P-type ATPases actually work. So let's begin with the general diagram, and then let's look at the specifics. So this is the lumen of the sarcoplasmic reticulum. This is the membrane of the sarcoplasmic reticulum, and this is the cytoplasm of the muscle cell. So we have a high concentration on the lumen side, a low concentration of calcium on the cytoplasmic side. So this is our P-type ATPase. In this case, it's the calcium ATPase. And notice it exists in a specific type of conformation. And the conformation basically allows the movement of these calcium ions into this region of that transmembrane protein. Once the calcium moves in, this uses ATP, it hydrolyzes ATP that creates a conformational change, and now it traps that calcium and it opens up to the other side, and the calcium can now move onto this side, the other side of that membrane. And so in this way, these types of P-type ATPases can basically couple ATP, so use ATP, the energy stored in ATP, and then drive the movement of these molecules to store that energy in the existence of this electrochemical gradient. Now, this is the general diagram of how these actually work, but what exactly is the structure of the calcium ATPase? So this is what it looks like. So we have four main regions. We have a transmembrane domain, and the transmembrane domain basically is found within that membrane. And we also have these three other domains, which are found on the cytoplasm side of that particular molecule. So we have A domain, which is this purple one. We have the P domain, which is this blue one. We have the N domain, which is this green one. Now, the end domain is called the end domain because it is actually responsible for binding that ATP nucleotide. So nucleotide begins with N, and that's why we call it the end domain. Then we have the blue domain. The blue domain accepts that phosphoryl group when that ATP is hydrolyzed into ADP. And so that's why we call it the P domain. And the A domain actually links that conformational change in these two domains to the conformational change in the transmembrane domain. So the A domain is responsible for stimulating the conformational change, as we'll see in just a moment. So let's actually take a look at the details of how the calcium ATPase actually works. And notice we have 6 different diagrams, and this basically cycles back and forth. So let's begin with the starting point, this diagram here. So this is known as the E1 state of this protein. So this is the calcium ATPase, the lumen side of the sarcoplasmic reticulum, the cytoplasm side of the sarcoplasmic reticulum. Now, in the E1 state, notice that this structure basically is open to this side of the cytoplasm. And 2, not 1, but 2 calcium ions can move into this pocket of the transmembrane domain. So it's the transmembrane domain that is responsible for actually binding those calcium ions. And it's 2 calcium ions per 1 ATP that is actually pumped to the other side of that particular membrane. So in the E1 state, we see that the ATP isn't actually bound, and these calcium ions can move into the transmembrane domain as shown here. And that's what happens when going from the E1 stage to the E1 calcium 2 stage. So now we have the 2 calcium ions inside the transmembrane domain, and now the ATP can actually go on and bind and interact with this green domain, the N domain. So remember, the N domain binds that ATP nucleotide to this section. And so now we go from this stage to the E1 calcium 2 ATP stage. And as soon as the ATP binds onto this green section, it creates a localized conformational change in the green structure, the blue structure, and this purple structure. And what that does is it seals off, it blocks off this entrance here and also exit. And so now these calcium ions are trapped inside this pocket, inside that transmembrane domain. Now notice this transmembrane domain didn't actually undergo a conformational change. Only these 3 domains underwent the transformational change, and that's what sealed off that exit. Now, in the next stage, we hydrolyze that ATP into an ADP, and that phosphoryl group is basically transferred onto the blue section, so this P domain. And so that P basically goes onto this blue section, the ADP remains attached onto the green section. And this stage is known as the E1P, where P is the phosphoryl calcium 2 ADP. Now, in the next stage, as the ADP begins to exit, so the ADP detaches, as it detaches, that creates a conformational change in the structure of that domain. And so what happens, that domain basically inverts. It opens up to the other side of the membrane, and that allows these calcium ions to actually exit that particular transmembrane region. And so now the calcium ions end up on the other side, on the lumen side of the membrane. And notice we still have that phosphoryl group attached onto the blue domain, and so that P domain. And so this stage is known as the E2P. And notice it's called E2 and not E1. So here we have E1, 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 and E1 because essentially this conformation is still open to the cytoplasmic side of that membrane. But the E2 means it is open to the other side, the lumen side of that membrane. So here we have this conformation, but here we have this conformation. That's why we have the E2. Now, in the final step, or I shouldn't say the final step, but in the next step, we basically use a water molecule to actually hydrolyze and remove this phosphoryl group from the P domain. And as it is removed, we basically form this E2 stage. And in the last step of this process, what happens is, as soon as that phosphoryl is removed, this interconverts back into the E1 stage. And so now, instead of being open this side, it is open to this side. When that takes place, this entire section basically opens up, and now these calcium ions can once again go into the transmembrane domain, and the cycle can basically restart itself. So we see that what the p-type ATPase does, what the calcium ATPase does, is it moves two of these calcium ions against the electrochemical gradient from a high to a low, from a low to a high concentration. And the way that it does this is by using the energy that is stored in these ATP molecules. So that's exactly why these membrane pumps are energy transducers. They essentially take one form of energy, in this case it's the energy stored in the ATP molecules, and then they transfer that energy and store it in the energy that exists as a result of this electrochemical gradient between the two sides